Okay, so here we're going to talk about the cell. I'm going to talk about the cell, parts of the cells, parts of structures associated with cells and tissues, such as collagen, as well as the replication, mitosis, meiosis. Now, just in general, you're going to have to know the general parts of a cell, which many of you already know. We're going to talk about differences between DNA, RNA, and proteins, replication, transcription, translation, again, kind of a review. We're going to have some discussions on ribosomal RNA, and then we're going to talk about the cell cycle and the mitosis and meiosis. We're going to bring in terms such as chiasma, um, and this is going to be important for diversity. So in general, here's a picture of a cell, and many of us are familiar with the cells and their structures. Um, Eukaryotes have membrane-bound organelles, whereas our prokaryotes do not. So in here, this is a eukaryote since we have our membrane-bound mitochondria, for example, or nucleus. We're going to have our Golgi apparatus. Some other important organelles are going to be our peroxisomes for fatty acid oxidation, our lysosomes to help break down things, proteins, digest things that are not functioning well. We're going to communicate between cells using junctions, gap junctions. We're going to have structure of cells with our microtubules. We're going to release things into the environment through vesicles. And we're going to interact with the environment through filaments, our plasma membrane, etc. So in general, we have DNA, RNA as nucleic acids. And DNA, of course, is going to contain the genetic information for life, and this gets passed on from next generation to next generation. And so fidelity is associated with the DNA. DNA gets replicated. And then we have RNA, which is going to be made from DNA. And RNA, which is important for making proteins, is messenger RNA. And so this is going to encode the instructions for building proteins, which are made up of amino acids. And we also have other RNAs that are not made for used to make proteins, but we have structural RNAs, such as ribosomal RNA. This forms a structure for protein production. We also have microRNAs, and these are going to be involved in gene regulation. We have other regulatory RNAs, and we have ribozymes, which are RNAs that act as enzymes. Our introns, for example, are ribozymes. Now, just as an example of genes that are involved in important cellular or tissue structure, such as collagen. Collagen is going to be an extracellular matrix. It is made by cells, so it is encoded in the DNA. And there are numerous proteins that are involved in the production of collagen through its various stages, from the nucleus to making our pro-collagen molecule all the way to our collagen fibril. So there are a plethora of different phenotypes with respect to defects of collagen, and it's because of all these genes that are involved. So we're going to have, of course, collagen molecule itself, which has the alpha chain, collagen type 1 alpha, and this is important for making type 1 collagen. We also have collagen type 1 alpha 2 chain, and this again is important for collagen. We then have a cartilage associated protein that's going to help process collagen. We have the LEPR1 gene, which is going to help process collagen as well. So we have genes that are actually structural, and then we have genes that are involved in processing, because collagen actually has a very dynamic process in which it goes through before it actually is released into the extracellular environment. So there are osteogenesis imperfecta, which is a collagen disorder, and most of these are going to be caused by defects of type 1 collagen, which is, of course, very important in bone. And the defects can cause bones to lose strength, to fracture, and it's going to lead to brittle bone disease. Now, this is also associated with tooth development, hearing loss, bone deformities. Stillborn infants are of type 2 osteogenesis imperfecta, and this is the perinatal lethal form. But here's just a table, and this is the first table. There are two tables here with that talks about difference in osteogenesis imperfecta. And so you have type 1, which is going to be a mild bone fragility, and it's that alpha 1 and alpha 2 that are going to have defects in. It also is associated with a blue sclera and hearing loss. Type 2 is the most severe form, and this is where you're going to have lethality in the prenatal or perinatal, and most of it's due to respiratory failure. And again, this is mutations in the same proteins. So you might ask why mutations in the same proteins can lead to differences in the phenotype. And it's just because you have different mutations within these proteins. And you're going to have a more severe loss of function of in type 2 than you would have in type 1, for example. And type 3 and type 4 are also associated with the alpha 1 and the alpha 2. We also have other osteogenesis imperfecta that are going to be involved in those processing proteins, or in alpha 1 and alpha 2 again. And so again, they're just different presentations based on the type of mutation. We have the 8, which is going to be similar to 2 in that it has potential for lethality. And so this is going to be associated with that LEPR1 gene, which is involved in processing of collagen. Okay, just really briefly, because this is just a very general overview. All genes that are going to, or all proteins that are going to be made from your DNA are going to be encoded in your DNA, and your DNA contains within it the start site of the genes. It has promoter regions of the genes. It's going to have enhancers upstream of the gene. It'll also have introns and exons. The introns are intervening sequences that get spliced out. The front of the gene will have a 5' cap. The tail of the gene will have a poly-A tail, poly-A adenylation site. Now, introns are going to be major portions of genes. We'll see in just a moment that a lot of our chromosome structure are introns, and so they tend to be much larger than the exons. And this is important because it helps protect those exons from mutations. Now, introns sometimes contain genes encoded within them that are unrelated to the gene for which the intron is actually in. So our neurofibromatosis type 1 gene contains 3 genes that are in opposite direction in the introns, and these 3 genes have no relationship to the neurofibromatosis type 1. So we have different types of DNA. We're going to have what's called single copy DNA, which is half of the genome, and part of the single copy DNA does encode proteins. DNA of the human DNA, only about 1 to 2% of the actual codons will encode for actual functional proteins. We also have a large portion of the DNA which is repetitive. So we're going to talk just briefly about these repetitive. We have satellites, dispersed repetitive. So here we have alpha satellites. You can see there's a big difference between the amount of DNA that's in introns, which gets spliced out of our messenger RNA, versus exons, which are actual coding regions for proteins. Now, again, protein coding regions are in our exons, and they're a very small fraction in comparison to our introns. Now, repetitive DNA are going to be called certain terms, and depending on their size mostly. So we're going to have these repetitive DNAs that are going to be scattered through the genome, and they do not typically occur what's called in tandem. They're going to make up a large percentage of the genome, about 45%. We're going to have some long interspersed elements or lines, and we're also going to have ALU elements, and these are able to make copies of themselves and insert themselves randomly in the genome. And these lines and ALU elements, if they do insert into a protein, they can disrupt that protein coding, and they can lead to a phenotype in disease. So while lines are long interspersed elements, we have our signs, which are short interspersed elements. And signs are basically 90 to 500 base pairs. You have in our genome a 300 base pair ALU restriction enzyme. And throughout the genome, there's about 1 million of these ALU repeats. So ALU repeats make up about 10% of the DNA, and they are within the sign repeats. Now, the long interspersed elements or lines are going to be about 7,000 base pairs, and these make up about 20% of the DNA. Now, satellite DNA is going to, they were given the term because of sedimentation in a gradient, and a gradient was made up of cesium chloride. Satellite DNA are going to be about 8 to 10%. You have alpha satellites, and these are going to have repeats of 171 base pairs, and they can extend up to 1 million base pairs. These are typically going to be found near the centromere, so the centromere is surrounded by repetitive DNA. There are also many satellites that are about 14 to 500 base pair long, and these are going to be variable between individuals. And then our microsatellites are 1 to 3 base pair longs, and these are usually going to be repeated over a few hundred base pairs. And these are also going to vary among people. And so these mini and micros are going to be important for typing, for example. All right, so let's just briefly talk about the cell cycle. So our cells are going to have the ability to replicate. And so we have, of course, our interphase. And our interphase is going to be made up generally of 3 different phases. We have our gap1 phase. We have our S phase, or synthesis phase. And we have our gap2 phase. Now, gap1 is where you're going to have synthesis of RNA and proteins. And the gap1 is where most cells will spend most of the time. And it could be hours, it could be years, or it could be always. So some cells never divide. And so if your cell does not divide, it is going to be gap 0. Now, gap 1 is where you're going to do normal metabolism. This is where you're going to make ATP, where you're going to do protein synthesis, etc. Now, if your cell is going to divide in the interphase, it'll move from gap 1 to the S phase, or the synthesis phase. And here's where you're going to actually have DNA replication. And then after DNA replication, you go through a gap 2 period where this is where the DNA can repair itself. It can prepare for mitosis, and the sister chromatids are going to be present here. Now, when it passes gap 2, it passes those repair mechanisms, it's going to move through mitosis. And mitosis is about 1 to 2 hours long to complete. After mitosis, the daughter cells will move back into gap 1, into interphase. Now for mitosis, you're basically going from a diploid cell to 2 daughter diploid cells. And so in mitosis, the phases are going to be prophase is first. So you move from gap 2 interphase to prophase. And in the prophase, your chromosomes will be visible. Your chromatids, your sister chromatids are going to be together attached at centromeres. And then our nuclear membrane is going to disappear. We're going to get our spindle fibers form, and these are going to be bipolar on opposite sides of the cell, and they will attach to the centromeres of the chromosomes and pull 2 sister chromatids to opposite directions. Now the next phase is metaphase. This is where you have the most condensed form of your chromosome, and this is where we typically do that staining so that we can see the chromosomes and see if there are any abnormalities. Chromosomes are going to be aligned in the equatorial plane, and the spindle fibers are going to pull the centromeres of the chromosomes. Now the anaphase is where your centromeres split. So the sister chromatids are separate. and they're pulled to opposite sides of the cells, and you're going to have 92 separated chromosomes. Now in telophase, is the final phase, is the formation of the nuclear membrane. Your spindle fibers disappear, your chromosomes decondense, and you get your cell division. Cytokinesis occurs. And then you're going to have two diploid daughter cells that develop from this. Now again, mitosis takes about 1 to 2 hours to complete, and meiosis is a type of cell division that is going to reduce the number of chromosomes in the parent cell by half. And instead of producing 2 diploid daughter cells, it's going to produce 4 haploid gamete cells. And so we're going to start with a diploid cell. And we're going to end with 4 gametes. All right, so in meiosis, we're going to have basically 2 meiotic divisions, and they're going to have the same steps and the same names that we just went through with mitosis. The difference is really going to be in meiosis 1. Meiosis 1 is the reduction division. Oops. This is the stage where you'll get 2 haploid cells from a diploid cell. And then meiosis 2 is going to be an equitational division. And you get each haploid cell divided. So let's just talk a little bit about meiosis 1. Now keep in mind that meiosis is a type of cell division that reduces the number of chromosomes by half. And this is a process that's required, of course, in our gametes, eggs, and sperm for sexual reproduction. And then this egg and sperm will eventually come back together and you'll have another diploid cell. Now in meiosis, similar to mitosis, the parent cell is diploid and it has 2 copies of each chromosome. So the parent cell in meiosis 1 is going to undergo 1 round of DNA replication, and then it's going to be followed by 2 separate cycles of nuclear division. And then you're going to get 4 daughter cells. Now meiosis is going to be similar to and different from mitosis. For example, in meiosis 1, you're going to have prophase 1, which is very complicated, which is very different than mitosis. You're going to have synapsis, which means homologous chromosomes pair up. This is unique in meiosis. You're going to have a chiasma form, and this is where you're going to get structures of homologous chromosomes coming together and exchanging material. So the chiasmas will cross over and you'll get exchange of regions of the chromosomes, so the swapping, and this is important for, of course, diversity. Now meiosis, the first meiotic division, is going to begin with prophase 1. And during prophase 1, the DNA and the chromatin condenses to form chromosomes. The pairs, known as sister chromatids, and they join at the center point called the centromere, and this is similar to mitosis. And then you get a large spindle that forms. Now between prophase 1 and metaphase 1, this is where you form tetrads of homologous chromosomes, and then the chromatid arms overlap, and this causes recombination. Now the end of prophase 1 is when you get bivalent chromosomes moving to the equatorial plane. You get your spindle forming and your nuclear membrane disappears. In metaphase 1, you have spindle formation, and you have bivalent chromatids attached by the chiasmata. Centromeres of the bivalents move to opposite sides of the equatorial plane. And then in anaphase 1, the chiasmata disappears. Homologous chromosomes are pulled to opposite sides of the cells. And the key difference is only half of the number of chromosomes migrate towards each pole, and the centromeres do not split. Now in telophase, you're going to get a new membrane forming, and each daughter cell is going to have the haploid number of chromosomes, and each chromosome has 2 sister chromatids. So you still have 2 chromosomes of each, but they're sister chromatids. Cytokinesis occurs just like in mitosis. Now, cytokinesis for sperm is going to be equal in meiosis 1. You're going to get the cytoplasm split equally. In the egg, all the cytoplasm is going to go to one daughter cell and the other becomes a polar body. Now, meiosis 1 is an overview. It's a reduction division. Prophase 1 is where you get crossing over. You get recombination between your chromatids, your homologous chromatids. Anaphase 1 is where your centromeres do not duplicate and divide, and only one member of each pair of chromosome migrates to each daughter cell. Now, in meiosis 2, you have no replication of DNA. The replication only occurs in meiosis 1. The interphase then is going to be very brief, followed by prophase, metaphase, anaphase. Prophase meiosis 2 is your chromosomes thicken again, nuclear membrane disappears again, spindle fibers form again. Metaphase 2, spindle fibers pull the chromosomes to a line at the equatorial plane. Meiotic anaphase is going to occurs same thing in meiosis 2. The centromere is split and a single chromatid is pulled towards each pole. Now, in this case, your sister chromatids may not be identical due to crossing over, so there is going to be diversity even at this level. And then they have telophase 2, which is similar to mitosis, cytokinesis, and you get 4 haploid cells. Now, for spermatogenesis, the sperm are going to be formed, of course, in the seminiferous tubules of the testes. You're going to have spermatogonia at different stages of differentiation, and the entire process takes 64 days. Now, for oogenesis, this is going to be prenatal. And at about the 3rd month of development, the oogonia of the embryo has begun to develop primary oocytes. You're going to have several million oocytes by the time of birth. Most degenerate, and only about 400 will mature. And these will be arrested in prophase for years until puberty, and then they'll begin to be released.